All right. Well, good afternoon, everyone. Julian, thank you very much for the introduction and, of course, for the invitation. So today, what I'll be doing is going back to a project that we have initiated about 2 years ago in order to resolve a gap in the field or an unmet medical need, which is to develop a cancer vaccine. So last time I presented some interesting data on our ARM vaccine, which is right now moving to clinical trial by the end of the year. So what I want to do is come back to this vaccine and give you some of the updates that we've generated lately on the generation of a second type or a second generation ARM vaccine for cancer immunotherapy. So again, for those that are not familiar with cancer vaccination, in fact, if we talk about vaccines in general, this is it could be achieved via different method or different modalities. You could use DNA molecules, RNA molecules, synthetic peptides, recombinant proteins. You can even use viral bacterial vectors to deliver a gene of interest, so it could be expressed in target cells. But of course, when we are really talking about cancer vaccination, the best approach we have so far are antigen-loaded dendritic cells. So why dendritic cells? Because these are the professional antigen-presenting cells in our body that have been generated for one purpose, which is to capture, process, and present extracellular non-self proteins to the immune system so it can see it and eventually attack it or attack the cell expressing it. And therefore, the hoped for outcome eventually is to take these antigen-loaded dendritic cells, put them back in patients, and then obtaining a T cell response. So that will be achieved by activating CD4 T cells, which will produce a variety of pro-inflammatory cytokines, driving the activation or supporting the activation and proliferation of the CD8 T cells, which at the end will end up attacking the tumor and destroying it over time. Now, this sounds pretty easy, pretty simple. This is the real knowledge we developed from this process. And if you go and you look what's out there that has been FDA approved or clinically used, well, the only cancer vaccine we have so far on the market is Sipuleucel-T, which is basically a dendritic cell-based vaccine manufactured by Dendreon against prostate cancer. So the way this vaccine works is that you would take, for example, monocytes or DC progenitors from the patient's blood. You would then activate it in vitro with a fusion protein composed of the PAP antigen for prostate cancer fused to GM-CSF. GM-CSF being a cytokine that drives DC maturation and differentiation to become an activated dendritic cell or antigen-presenting cell capable to activate both CD4 and CD8 T cells, hoping eventually to kill or attack prostate tumors. Although preclinical data have shown fantastic results, the problem is when this vaccine was applied in the clinic, it did not really lead to a long-term or long-lasting effect. In other words, patients were paying something like $100,000 to only get 4 to 6 months improvement in survival. So what that means is that there's an urgent need to develop effective, long-lasting anti-cancer vaccines. We're far from there. People have been using CAR T cells to really destroy tumors, but that's only specific to one tumor type, which is B-cell lymphoma or leukemia, targeting always the same antigen. So the idea is, how could we go from the CAR T cell technology to develop something that not only will kill cancer, but will give us also a memory response? That's really important, and that's the basics. That's the basis of the cancer vaccination, which is to develop a memory response to be able to protect the host from subsequent relapses that can occur with time. So where does defense therapeutics fit in all this? So theoretically, if you want to develop any kind of therapy, whether it's DNA, RNA, protein-based or antibody-based, all of these products will end up being endocytosed by antigen-presenting cells. So what that means is that this extracellular product will be captured into a structure called the endosome. And what the endosome has as a single purpose is to actually degrade these products into smaller portions that will enable them afterwards to be secreted or pass on to the cytosol and eventually be used by the cell. So if you can imagine you have an antigen or a protein that you would like to immunize against, well, that protein will be non-specifically damaged by the low pH, so the high acidity in the endosome and the activated proteases, which might destroy the immunogenic peptides or the immunogenic sequences that could be highly beneficial for cancer treatment. And so what defense is doing is that they're developing technologies to overcome these molecular barriers or molecular limitations by really allowing this technology to be adapted to a variety of different products, such as vaccines, antibody-drug conjugates, and even the development of other types of biomedicine. So how do we do this? Well, the technology platform that Defense is developing or working on is the Acume platform. So it's a small molecule composed of a bile acid and a nuclear localization signal. But as you can imagine, these two components could be interchanged in the sense that you can have any kind of bile acid out there. You can have different peptide sequences to generate what we call variants of the Acume. And what this molecule does is that actually it breaks down the endosomal membrane, allowing the product you're using to move or escape from the endosome to the cytosol. So in this case, you're really making sure that the endosome is not maturing or not not destroying the product that has been captured, and therefore your product will end up in the cytosol in an intact form, which could be subsequently used effectively by the target cell. The best example, and that's how I like to show this slide all the time, is to give you a visual example of how the Acume works. So if you take, for example, a dendritic cell, and we know that glycosylated proteins, so proteins that have sugar residues, are always on the cell surface. And if you have the endocytosis happening, you end up having those glycosylated protein inside the endosome. So that's why it shows you here white and white in both conditions. So if you overexpress, let's say, a fusion protein composed of the human galactine-3, so that's a protein that binds to carbohydrate sugars, which is fused to GFP, so the protein will look like green, then you can see that the entire cell is green, meaning that you have a natural distribution of the fusion protein all over the cell, non-specific distribution. However, if you stimulate that cell or you pulse that cell with the Acume molecule, then what happens is that the endosomes are damaged. And when they are damaged, this fusion protein now can move in and out of the endosome freely. And that's why you go from a completely green cell into a cell where you have endosomal structures on puncti of green, which represents endosomal structures that have been damaged, which have accumulated this fusion protein. So that's a very neat way of showing you that the Acume really destroys the endosomal membrane, allowing products to go in and out of the endosome. Of course, we've done a proof of concept. We know that DC vaccines have failed for many reasons. Could be manufacturing, but could also be a problem related to endosomal acidification and destruction of the antigens. So we've actually demonstrated that the Acume can enhance significantly, I'm talking here 50 to 100-fold, the therapeutic potency of dendritic cells, a study that was published in Cell Reports Medicine. And we've also published a review summarizing really how the Acume technology works, not only in the context of vaccines, but really in antibody-drug conjugates and how we can exploit it for other applications. So if you're really interested to learn more about the Acume, I encourage you to go read that review, the paper, and we have more studies coming on this year. So the Acume technology so far has been applied to different verticals. So initially we thought that, we're going to develop it in the context of antibody-drug conjugates. But then when we understood how the technology works, we found that this versatile approach or the versatile technology could be applied to the development of protein-based vaccines, cell-based vaccines. You can tag it to antibody-drug conjugates, develop your own antibody-drug conjugate, or you can even modify it to come up with a molecule that has novel anti-cancer properties. And we've got examples of each one of these different verticals or different platforms. However, what I'll be doing today is really talk to you about one of our lead cell vaccine that is really moving right now to phase 1 trial. So this slide here represents the different pipeline we have right now. So as I mentioned to you, we've got small molecules, we've got protein vaccine for cervical cancer, antibody drug conjugates in development, mRNA vaccines, but we've got also the cell vaccine, which we call the ARM, for A1 reprogrammed mesenchymal stromal cells. So that's what I'm going to be talking about today. So why did we decide to move from DCs and focus on mesenchymal stromal cells? So mesenchymal stromal cells have been known for over now 50 years. So they've been reported or discovered in the 1970s. And one of the first application for these cells was their use in the field of regenerative medicine to really to actually repair any ischemic area, destroyed area, damaged tissues. And in fact, if you do a PubMed search, you'll find there's more than 6,500 studies where people have been using MSCs to repair target tissues. On the other hand, because they have a very neat secretome profile, so they have a lot of chemokines, suppressive factors, and cytokines, they've been also used in the field of immune suppression. And there's more than 700 studies reporting their use in all kinds of autoimmune disease, arthritis, multiple sclerosis, GVHD, and whatnot. And so why do people really love MSCs? Because these cells are easily harvested. You can have them from the bone marrow, from adipose tissues. You can use them from the umbilical cord. You could actually expand them very easily in vitro with a chemically defined milieu or condition. I mean, media that doesn't require cytokines like in the context of dendritic cells. They've been, of course, exhibiting a very nice safety profile, and that's coming from over 500 clinical studies where patients have been receiving MSCs, and we haven't seen or observed any side effects. And they have this beautiful plasticity, meaning that you could basically push them the way you want. They could be immune suppressive, they could be immune stimulatory, they could be anything, and therefore they could be adapted to any problem you're trying to resolve. And that's where we came into play, because we said, okay, well, everybody's focused on these two different fields, but can we basically exploit MSCs to vaccinate, to induce an immune response? And here we have different examples where we've demonstrated that we could basically overexpress proteasomal genes to make them antigen-presenting cells, whether it's the immunoproteasome, the thymoproteasome. We can pharmacologically stimulate them or activate them with drugs to make them behave as antigen-presenting cells. So there are multiple examples of how we can basically convert these MSCs into antigen-presenting cells. And therefore, when I saw this and I saw the Acume technology, the first thing I asked myself is, can we somehow play with the Acume molecule and come up with a variant that could convert MSCs into antigen-presenting cells? And that's the idea we actually initiated 2 years ago, where we said, okay, we have a system that would allow us to screen for molecules that are able to induce antigen cross-presentation. Antigen cross-presentation means you capture an antigen from the extracellular environment, you process it, and you present it on MHC molecules to responding T cells. And we have a nice assay here, as shown in this schematic diagram, demonstrating that the B3Z T cells could give us a enzymatic reaction whenever it's interacting with MHC1, presenting the synfical peptide, which is derived from the ova protein. So it's just a peptide that is, after processing, being presented on the cell surface. So of course, we took different Acume variants. We mixed those with the ova antigen, we did the assay, and we discovered the A1 variant here, which is able to really induce a nice activation of those B3Z. So using that A1 variant, we then, conducted a lot of in vitro studies. But one other thing we wanted to understand is, how would the A1 convert mesenchymal stromal cells into antigen-presenting cells? And so when we did a transcriptomic study, so we stimulated the cells with the A1 with or without the antigen. We took the RNA molecules, we sequenced all of them to see what are the genes that are being downregulated and upregulated. There we noticed that there's more than 50 and 100 genes that are really modulated. So you've got genes that are upregulated and downregulated. And when we looked using bioinformatic tools to some of the processes that were somehow induced by these ARM cells, we were actually very happy to notice that there is a so-called unfolded protein response. So the unfolded protein response is a process where a cell would feel something is wrong at the protein level. So there are probably misfolded proteins or probably protein aggregates that are being formed or captured by the cell, and these are naturally toxic for the cell. So what the cell does to respond to these protein accumulation or aggregation is that it turns on the unfolded protein response as a means to basically activate genes and molecules that can somehow rescue the cell from cell death. It does so by inducing chaperone proteins, which are there to really unfold or repair the folding. But one of the most important processes that the unfolded protein response does is that it activates degradation by the proteasomal component. So what that means, and that's really just a summary of how the A1 molecule works. So when we mix the A1 molecule with an OVA, or the protein that we're using, these will form protein aggregates. So these protein aggregates will be captured by the endosome, and therefore, the ACUM will then activate NOx, which is a protein responsible for producing reactive oxygen species. Those reactive oxygen species will then induce what we call lipid peroxidation to break the endosomal membrane. That's what the ACUM usually does. And that allows really the protein aggregates to be released into the cytosol. Once this antigen release happens, so now the cell feels that there is protein aggregates, it turns on the unfolded protein response and targets the antigen that were captured and released in the cytosol for degradation by the proteasomal machinery, which eventually leads to antigen presentation on the cell surface. So I'm here summarizing 2 years worth of work in one slide. But what I want to really bring your attention to is the potency of this vaccine. So one of the first questions we have to ask ourselves is, and this is beautiful, it's actually happening in the cell, but would basically this ARM vaccine induce a very good potent therapeutic effect in vivo? So of course, we've conducted over 10 different in vivo studies looking at routing of administration, the dose, syngeneic versus allogeneic. And here I'm showing you one of the key study in vivo showing how the vaccine is highly potent and effective. So what we did in this case is we took immunocompetent animals. We transplanted subcutaneously a T-cell lymphoma solid tumor expressing the ovalbumin antigen, just an experimental antigen. And then we, 2-3 days after, so after the tumor has been established, we vaccinated these animals with 500,000 ARM cells delivered subcutaneously. So they received 2 doses of the vaccine in combination with the immune checkpoint anti-PD-1, which is the standard immune checkpoint used right now in the clinic. And in this case, the vaccine was basically an allogeneic preparation, meaning it's not coming from the same genetic background. This is the equivalent of saying we have a donor that is unrelated to a patient. He delivers, he gives bone marrow samples. The MSCs are expanded from that donor. They're converted to ARM cells, and then they're injected into a non-compatible patient. So that's an allogeneic universal vaccine. And here what we noticed was the following. So of course, we've got all the controls. So you see how the EG7 tumors grows very quickly in these animals. If you put, for example, regular MSCs pulse with OVA, not that much happens. If you take OVA-stimulated MSCs and you add the PD-1 immune checkpoint, you could see some sort of a delay, but nothing really major. Now, if you take the ARM vaccine, so the A1 plus OVA, and then you pulse the MSCs to get the vaccine and use it as a monotherapy, you can notice how nice is the control that is happening for over tumor growth. So, of course, we've got at the end 60% survival, so 4 mice died out of 10. But remember, this is a monotherapy, and not a lot of monotherapies actually do work. Monotherapies usually fail. They could delay tumor growth, but they end up failing. But now if you combine it with the immune checkpoint, which is what we think, what we're going to be using in the clinic, there you can see that the control of the tumor growth was very efficient to the point where we got a 90% complete response, meaning 9 out of 10 animals had their tumors gone. So that was really impressive data. And we've actually repeated that same experiment in the melanoma model. And instead of using the ovalbumin protein, we've used basically tumor lysate. So this is how we're going to be using it, using the technology in our clinical trial. Now, over the years, we've noticed one very important point, which is stability of the A1 monomer. So as I mentioned to you, it's composed of a bile acid, a specific peptide, and this molecule was synthesized, was stored -80, was used back and forth. But then we noticed that after a month or two, we're not getting a consistent activation or generation of the arm, and that's normal. You know, drugs or small molecules get degraded over time. So we started inquiring and working with our chemists. And what we ended up developing is an A1 dimer. We found that if you basically fuse two A1 molecules together, you enhance significantly, in fact, very efficiently, the half-life of the molecule. And of course, when we confirmed the potency of this A1 dimer to induce antigen cross-presentation by MSCs, we noticed here that 20 micromolar of the A1 dimer was generating an antigen T-cell response equivalent to 50 micromolar of the A1 monomer. So we decreased the dose by about 60%, and we couldn't go higher because we noticed after that the cells were dying. You know, the monomer was so active that it was causing so much protein aggregation and end up killing the cell. So we had to find the right dose that makes our cells work effectively. So, of course, we identified the dose in vitro. The next question is, does it work in vivo? And the answer, I mean, to answer that question, we repeated the exact same study that was actually done in our in vivo experiment shown in two previous slides. And when we did that study, we noticed in this case that we got, again, 100% complete response when the second generation ARM vaccine was used in combination with the PD-1 immune checkpoint. So that was really interesting to us. And now we're thinking now to basically re-challenge those cured animals with the same tumor to demonstrate that we have generated a long-lasting memory response that not only initially destroyed the tumor, but can protect the host from subsequent challenges with the same tumor. And of course, these animals are all live as we speak. So I've been talking to you about the phase 1 trial since the end of last year. In fact, we have started the manufacturing of our vaccine at the Cell Processing Center here in a hospital affiliated to McGill University. So right now, the clean room is conducting manufacturing runs from 3 different donors to be able to confirm manufacturing consistencies in order to present the file to Health Canada and get the approval to initiate a phase 1 trial against solid tumors. So the point here is to use the ARM vaccine against head and neck and different types of melanomas in patients as a monotherapy and as well as in combination with Keytruda to demonstrate not only safety but some efficacy data to be able to move on to a phase 2A. So overall, here's a summary of the presentation today. So basically, this is another example demonstrating that it is indeed possible to convert mesenchymal stromal cells, which are initially known as immunosuppressive cells or anti-inflammatory, and that's why they're used to repair tissues, into potent antigen-presenting cells. So we can convert them into a cell that could trigger T-cell response. These ARM cells can effectively cross-present antigens. In the previous presentation, I showed you data that these cells could capture more efficiently the antigen, process way more efficiently the antigen, and that explains why we're able to present it very efficiently on the cell surface and activate T cells. Mechanistically, we now can say that A1 induces protein aggregation, which ends up turning on the unfolded protein response in target MSCs. And as a result of this, these MSCs would then activate the antigen degradation by the proteasome, present the peptides, and activate T cells. So the ARM cell or the ARM vaccine could induce tumor regression, both with the first and second generation, but that happens really when you combine it with the immune checkpoint inhibitors, such as PD-1. But we notice also that the A1 monomer is somehow unstable over time. And this is why we developed the A1 dimer, or the second generation A1 molecule, which was tested and confirmed to lead to the same therapeutic effect. And finally, the arm cell vaccine is currently being manufactured in order to get the approval by the end of this year to start a phase 1 trial in patients. So with this, I'd like to thank you all very much for your time and attention, and please feel free to communicate with us if you have any questions or you'd like to know more about the ARM technology or other programs, and I'll be more than happy to take further questions. Thank you.
Well, thank you very much for the great presentation and update, Doctor. So maybe one question. When we see such research being done, what is the actual, the next phases and the development time from which you have those great results and then we can expect commercialization and maybe even saving lives up to there?
Yeah, so great question. So right now we are at the point of manufacturing. So that means we're trying to demonstrate to Health Canada, which is the regulatory body equivalent to the FDA, that we can generate doses for patients very effectively. We're showing the timeline, which should be within a period of 2 to 3 weeks. And once we get the dry runs completed by, I want to say, the end of the summer, the file will be submitted to Health Canada to be able to initiate a phase 1 trial. So the phase 1 trial usually would last for about 1 to 2 years, depending on the therapeutic objectives or the objectives overall of the trial. But the idea really is to start seeing some efficacy results, not only that the vaccine is safe, which we expect will be safe, but also to demonstrate that this vaccine is indeed capable of inducing regression. So that will allow patients that have failed, let's say, first, second, or third line of treatments to really enroll in the clinical trial and start seeing some good effects on them. Now, I know that things are progressing very rapidly. So in Japan, for example, if you finish a phase 2 trial and you show some very nice efficacy data, there is actually the opportunity to start treating patients and getting reimbursed, although the phase 3 trial has not been completed. I think this is something that North America is looking into it. But overall, the timeline for developing this vaccine would be between, I want to say, 7 and 10 years if you want to really count the phase 1, 2, and 3 trials. Having said that, you know, these trials are open and eventually they can enroll patients that have failed their therapies.
Good. That gives us a good idea. And then how much is the cost of development for such vaccines on those 7 years, for example?
Yes. So if you know, a phase 1 trial would be in the range of 5 million Canadian dollars if you really want to make sure that you complete it from A to Z. You want to move on to phase 2, we're talking about 15 to 20 million. And of course, when we talk to phase 3, that's where we would like to partner eventually with a large pharma, because here the cost will be pretty large, because we'll have multi-centers, different countries involved, and that will be requiring a lot of financial support. But what's really important to mention here is that we believe that this vaccine would cost 1/10, if not cheaper, than the current cost for the CAR T dose, which could be, which is really, I would say, between $400,000 and $500,000 at the moment. So our vaccine would be in the range of, you know, 10 to 20K per dose, which is really negligible, knowing how the Sipuleucel-T is in the range of $96,000 US. So, but of course, for cancer, as a cancer vaccine, that cost is really, really small, and with benefits that it will be inducing at the end.
Perfect. One last question before we go to our next presenter. Maybe if you can emphasize on something that's very important. So the Acume can actually obviously work as a platform. Now, what you're searching and the ways that you're currently exploring, are those everything that Acume can does, or those are the one that you specifically chose in order to achieve specific goals, for example?
Well, to be honest with you, Acume is astonishing. It's actually given us surprises every time. Of course, initially in 2017, when the company was created, the objective was to apply Acume to antibody drug conjugates. But now we know that this is not the case. You could use it to convert cells, you know, dendritic cells, mesenchymal stromal cells, other cell types, probably like B cells, into antigen-presenting cells. You could use it for ADCs. You could now we're developing an mRNA vaccination program. So we not only we're focusing on cancer, but now we want to start a malaria vaccine program where we have an mRNA encoding for multiple antigens, so we can deliver these effectively. We're thinking to really partnership with Moderna and BioNTech to really develop their own mRNA cancer vaccines. We've got the acute molecules on their own can become anti-cancer molecules. So that's another field that we've spoken about like last year with the AcuTox starting a phase 1 trial in the US by the end of the year. So there are multiple avenues, and I'm pretty sure if we continue digging more, we're going to find new applications for the Akim technology.
Perfect. That's great. Well, obviously wish you the best of luck in your quest. Obviously, a lot of people would like those solutions to be already available by now, but one thing is sure, they can keep following your story. Write them if you have any questions. It is absolutely interesting. And thank you very much again for the very interesting presentation.
Thank you. Thank you for the invitation. Have a great day.
A pleasure. Thank you.
Bye.